Good evening, children. Welcome to another practical assignment. Today's practical is practical number 8. As usual, we'll start working on a new sheet. Today's date, 19th of August, in one corner. The aim of experiment is to study osmosis with the help of a potato osmoscope. The requirements are very simple. A large potato, pins, petri dish, 20% sugar solution, water, knife, or a scalpel. See, the idea is to use a potato and we will make a flat edge of it. We'll cut it with a knife, we'll peel off the potato and make one end flat and let it stand into a petri plate, right, like this. The outside will be filled with normal plain water, but inside we will make a cavity out of the potato with the help of knife, and 20% sugar solution or a concentrated sugar solution will be placed. So this will be left for some time and the level here will be marked with a normal pin. Now, when the observation is made after some time, we will see that the water has gone inside the cavity and the level has risen. So, what has happened over here? The process of osmosis has taken place. Remember, osmosis, the movement of water molecules from the region of higher concentration, that is outside, to inside, that is to the region of lower concentration, through a semi-permeable membrane. Right? So this is what you're going to show in this experiment by very simple working. So let us see what is the procedure. Take a large size potato tuber, cut its one side so as to make it flat. Bore a cavity from the other side in such a way that a very thin base is left intact on the flat side. Remove the skin near the edge of the flat end because skin of the tuber is impermeable to water. So this is why we have to peel it, because the skin of potato will be impermeable and we have to remove the peel. Pour 20% sugar solution in the cavity of the tuber up to 1/2. So we'll up to almost 1/2. We'll pour the sugar solution inside. Mark the level of sugar solution in the cavity with the help of a pin. Place the tuber on its flat end in a petri dish half full of water. So this petri dish also should be almost half full, just like half full potato osmoscope. Note that after some time, the level of sugar solution has risen in the cavity. Here it will rise, whereas now later we'll see the level in the petri plate slightly falls but rises inside. Mark this level or the reading also with another pin. So later we'll mark the final reading also with another pin like this. Observations. The level of sugar solution in the cavity of the potato tuber rises, indicating that the solution has absorbed water from the petri dish. The two are separated from each other by large number of cells of the tuber. The entry of water into the sugar solution therefore proves that sugar solution is osmotically active. Second, the cytoplasm of the cells of tuber that lies between the sugar solution and the water of petri plate acts as a single semi-permeable membrane. And third, water enters the sugar solution when it is separated from it by semi-permeable membrane. This process is called osmosis. Remember, children, osmosis has to, without semi-permeable membrane, osmosis cannot occur. So semi-permeable membrane is a must for the process. After this, we'll write definition. Osmosis is the movement of water molecules from the region of their higher concentration to the region of their lower concentration through a semi-permeable membrane. Then we have certain precautions to write. Number one, the cavity should be deep so as to leave only a thin layer of tissue as at the base. So we'll make sure that the cavity is deep enough to just leave a layer of tissue at the base. Second, remove the skin of the tuber from the base and the sides. That is, from the base also, whereas from the sides also, we will remove the skin. Third, make the base flat so as to keep the tuber flat and stable in the dish. And fourth, sugar solution should be hypertonic, that means concentrated, as compared to the cell sap of the tuber cells. Importance of osmosis. These are the generalized points we're talking about. Number one, it helps the plant to absorb water from the soil. Second, it helps the movement of water from one cell to another in an organism. And third, it helps in many types of plant movements like the folding and drooping of leaves. In short, what we call as wilting, right? Opening and closing of stomata, etc. So, the work is simple, children. You need to write down starting from here, from the heading practical number or experiment number 8, the aim, requirements, procedure, observations, and then definition, precautions, and importance. So, this is all the writing part that you have to write in your files. And the diagram, you will draw both the diagrams, that is the initial and the final level. You should be very carefully drawing their diagram, making sure that the initial level, right, here the level outside and inside should be almost at the comparable average level. Then the final level inside rises and the outside level slightly goes down. Don't make a radical difference of levels because the change is slight. So that's all with the original level and the final level that you need to draw. So today's assignment I'll be posting on 19th, and children, you need to submit the work by 22nd. So that's all for just now. Thank you.